Okay, welcome. This is a special recording to get me caught up in lectures because I've got a little bit behind on the lectures for 303. So we're going to finish up the last 30 slides of the genetics and genomics lecture this way. And we ended up looking at changes to DNA sequences and mutations. And we wanted to talk more about it going forward. So here we go. So you can have errors during replication we talked about. So about one nucleotide in 10 to the 8 is incorrect. So that you can get one mutation every 100 million base pairs. Spontaneous chemical changes in bases, you can have them change, like a deamination of cytosine to produce uracil as an example. Also DNA damage agents. These are mutagens that damage DNA, and this is how often we think about how mutations might happen. All of these can cause mutations, and if the daughter cell inherits that change, it is a mutant. So these uncorrected errors produce genetic change in daughter cells. Now, this example here shows the wild type, and you have GC base pair right here, and then a T is mistakenly put in. If DNA replication occurs before this is repaired, you now have a mutant daughter cell with an AT base pair instead of a GC base pair. All right, so what are the types of chemical mutagens you can have? Well, there are base analogs. These are 5-bromouracil and 2-aminopurine, for examples, and they mimic certain bases. So 5-bromouracil will mimic both an A and a G sometimes. So it incorporates like T and occasionally falsely pairs with a G. 2-aminopurine incorporates like an A and sometimes does a faulty pairing with a C. So in this case, you get an ATG to a GC, and you get these changes in bases. There's also chemicals that react with the DNA, like nitrous acid and hydroxylamine, and they deaminate A's and C's or reacts with the C, and again, these have to be repaired, and then you get these point mutations. There's alkylating agents that put on alkyl groups, and they cause point mutations and deletions. Intercalating agents will cause microinsertions and microdeletions, and these end up in causing frame shifts. And then radiation. Now these are and again, radiation will cause damage, and that the repair may lead to errors. Now this is for illustration only. No classes and mechanisms, but you don't need to know specific chemicals. Cells can repair damage in different ways. Here are a list of different types of repair systems in the cell, and most cells will have these repair systems. Probably the most common is the mismatch repair system, here called methyl mismatch, and this one will repair 99% of mutations. There's also base excision repair that will take out a region and take out the base that mispairs and then put it back so it will fix modified bases. This is very accurate. Nucleotide excision repair, this will take out the whole nucleotide, not just the base. Again, this is very accurate. Photoreactivation repairs pyrimidine dimers, TT dimers, CC dimers that are caused by UV light. And this is again very accurate. You then have recombination repair. This can fix single-stranded large gaps in the DNA, and this by recombination, this is very accurate. And then you have translesion bypass synthesis. It's also known as the SOS repair system. It's part of SOS system, and it's error prone. It generates mutations, and we'll talk about that. Finally, there's one more that isn't mentioned there, and that's methyltransferase. And this is actually a protein that scans the DNA, runs down, and if this enzyme finds an inappropriate methyl group, it will remove that methyl group and fix the DNA and put it on itself, and this actually inactivates the protein, so it's not really an enzyme at all. And this whole protein is made just to get rid of methyl groups. Let's go through some practice. Hopefully you've read the genetics and genomics chapter, talked about these different repair systems. Let's see if you can figure out what caused mutation and then how you'd fix it. Okay, first one. What probably caused this T to G mutation? Shown at the bottom here. Okay, hopefully you've made your decision. The correct answer is probably replication. Right? So you have this base made. How would the cell fix this? Now the easiest way to fix this is, pick one. And the correct answer is mismatch repair. Remember, that fixes 99% of errors, so if you're not sure which one, pick mismatch repair. Another thing that you can have is frame shift mutations. These occur in redundant runs of bases. As the replication fork is passing, one strand slips versus the other, and a base can be added or deleted because of this. Many of these are fixed by repair mechanisms. Most often mismatch repair is shown here. So you see this extra base is put in. Obviously this bubble is recognized by the repair systems. If the repair systems get there first, you'll get this fixed. Everything's fine. If the repair system doesn't get there in time, this gets replicated. You have one normal chromosome, but then you have one mutant chromosome where an extra base has been put in. And of course, as we've talked about before, these type of frame shift mutations are quite deleterious. Real quick reminder, here's a mutation that you see. What could have caused this? Okay, the answer is the same again. It is a replication error, as we just talked about. Now, how would you fix this? And if you picked mismatch repair, you are correct. Okay, let's go on to another one. What probably caused this mutation? Okay, this mutation was caused by UV irradiation. How would you fix this mutation? Okay, the correct answer is B, photo repair. Here's another one. What probably caused this? And the correct answer is methylation. And how would you fix it? Again, this is the enzyme methyltransferase that would come in and get rid of that. In an exam, I hope that you'll be able to figure out what causes mutations and then what system is used to repair them. So, here's another one. There's a block in the DNA where bases are damaged and the other strand is missing. How would the cell fix this? Well, it could use either recombination repair or SOS repair. So let's look at some examples. So here's the DNA lesion, right? It comes in and then you get a demise of the replication fork. The replication fork is coming along and it can't really replicate through this area because there's so much damage in the DNA. The bases are different. There's a large stretch where there may be missing bases or whatever. So what happens is the cell will actually, Rec A will come in and it will, the replication fork will regress past the damaged area. Nucleases come in and then degrade the damaged area, and then you borrow the replicated strand on the other side. So you go back through, you see you've replicated over here already, you go back through, you take this strand, you bring it over here, you then copy it, and then you resolve the strand and continue. This type of recombination repair is incredibly effective. Now, we've talked about the REC system before, the homologous recombination system before, when we talked about horizontal gene transfer. This is what this system is really designed to do. As a sidelight, it will sometimes incorporate foreign DNA, but it's really a repair system, and it's for repairing serious damage in the DNA. Now, if all other systems fail, for example, you have single-stranded DNA, one of the strands has been lost, and then you have damage in the DNA, and this is really serious damage, so there's no base pairs there, all sorts of messes going on, you really don't know what to do, this system will come in, and it's the SOS repair system. It basically, it's a last-ditch effort. So here's the polymerase coming on, bam, it hits this, DNA polymerase then exits, Rec A binds, and then this other DNA system comes in. This is polymerase 5. It's UMUC-D complex. This comes in, blasts through this area that has the damaged area, and then the regular DNA polymerase comes back through. Basically, it's just a way of getting this replicated and even leaving the damage there when you have no other option. And why would you do this? You've got no other choice. If you don't replicate this DNA, you're dead, so you might as well try and replicate it some way and get it fixed. SOS repair is a system that causes lots of errors, and it's a last-ditch effort. It's not what you want to use. So that summarizes the types of errors that you can have in the DNA. You can have replication errors, and these replication errors can cause base pair substitutions. They can cause frame shifts. You can have radiation and other things causing breaks in the DNA, single-stranded DNA, other disruptions. You can have UV radiation causing dimers, pyrimidine dimers, these TT dimers. From that, the cell has a huge number of repair mechanisms. And in most cases, these repair mechanisms will fix the problems with the DNA. However, every once in a while, extremely rarely, a mutation gets through. And the question is, what is the effect of that mutation? So let's define a mutation again, just to remind you. A mutation is a change in the genotype of the organism. And again, the genotype of the organism is the nucleotide sequence of the genome. The question is, does it change the phenotype of the organism? And the phenotype is the observable properties of the organism. So the effect of a mutation can be neutral, detrimental in many cases, or beneficial in rare cases. The effect of the mutation depends on what the DNA encodes. Mutations in coding sequences can cause a loss of function, or what's called a knockout mutation, where the enzyme or protein is completely non-functional. It can alter the function of the protein. It can decrease its activity. It can increase its activity. It can make it not respond to any allosteric effectors anymore, etc., etc. Sometimes it can cause a gain in function. Maybe the enzyme can now do something it couldn't before. It can recognize a slightly different substrate, etc., etc. You can also have mutations in gene control regions, such as promoters, other binding sites for regulators, etc., etc. And this can change the pattern of gene expression, and that can be huge. It can be really important sometimes. So more about the types of mutations. Point mutations, insertions, deletions small, insertions, deletions large, inversions, which is just bases turning around, complete rearrangements and duplications. We're not going to talk too much about these because they're very complex and they're hard to detect. You can learn more about that in Micro 470 if you want. Okay, now, remember there can be silent mutations, and we've talked a lot about these, so I'll kind of blow through this. Silent mutations don't change it. You change a base pair, the base pair codes for the same amino acid, doesn't change it. You can have point mutations. They can sometimes cause a single amino acid change. Sometimes this can be significant, sometimes it's not. Non-send point mutations normally are significant, as they'll change a coding codon for a serine here to a stop codon, and that will stop the protein. That can have all sorts of deleterious effects. Clicker question. Silent point mutations are most likely to occur in the first nucleotide, the second, or the third. And what we've talked about in the central dogma, you know it's the third nucleotide of a codon. Again, just a reminder, note the wobble position is the third position, and this often codes for the same amino acid. So here you can see all of these code for proline, all of these code for threonine, etc., etc. It's even cooler in that if you look at leucine and isoleucine, they're lined up here. So you can actually have changes in the first or third, and you'll end up with a similar amino acid inserted. So code's arranged in a way to make it very robust. Again, we've talked about insertion mutations, so I'll just breeze through this very quickly in that, you know, if you insert bases in here, you'll completely change the frame of it if it's 1 or 2 bases, and that again will completely change the frame, will completely change the protein, and most often cause a knockout. Same thing with the deletion frame shift. Okay, clicker question. Which mutation is most likely to affect protein function? And I'll let you read these off. Okay, this one's a little tougher. The correct answer is B, a deletion of 5 nucleotides in the middle of a coding sequence. In A, the missense mutation is just changing a leucine to an isoleucine. Those are very similar in structure, so that wouldn't be it. A nonsense mutation in the last couple of codons isn't going to cause a problem, because if you're just eliminating those, it would probably just be fine. And an insertion of 3 nucleotides at the beginning of a gene, you're just adding an extra amino acid. That could cause problems, but it's much more likely that B, a dilution of 5 nucleotides in the middle of a coding sequence, would cause a problem. Okay, that ends this section, talking about mutations and their consequences. Now we're going to move on to how do we detect those mutations when we're doing genetics. So let's imagine that you have a bacterial culture and you're interested in finding out which of them are resistant to rifampicin. You're studying that drug resistance for some reason. So you mutagenize it in some way. Let's say you expose it to UV light. You then plate this on a rich medium containing rifampicin, such that you're demanding that everything that grows on this plate must be a rifampicin resistant colony. This is a selection. You are selecting directly for the type of mutation that you want. Conditions are set up such that only the mutants of interest are able to grow. And then this is very powerful, and if you're doing any type of genetics, you always want to try and design selections. In a lot of cases that isn't possible, so then you have to go on to screens. In a screen, most cells grow with the desired mutant being detectably different in some fashion. You normally use screens when you can't really design a good selection. And there's all sorts of types of screens, and we're going to go through them here. There are assays of an enzyme, replical plating, color indicator plates, and then hybridization. So an enzyme assay, this is a brute force approach. You again mutagenize your culture, let's say with a chemical agent this time. You then dilute it out and you plate them all out. You make thousands of colonies, and you pick each of those thousands of colonies and you put it into a culture and grow it. And then you grow all those thousands of colonies and you assay for an enzyme of interest. Let's say you're interested in knocking out a certain enzyme. You find the one that's not able to do that enzymatic activity and you save it. Or let's imagine you're looking for antibiotic increase in production of an antibiotic. So you measure the amount of antibiotic all these guys make, and you find that this one makes more, so then you grab that mutant and you go on. This is actually how antibiotic producing strains, they increase production in industry, in the pharmaceutical industry in a lot of cases. Obviously, this is a very brute force approach. It's very laborious. Modern day robots that you can have made this process easier, and that you can have the robot do a lot of these procedures. And it's an effective method, but it does take a lot of effort. Another example is replica plates. So in a replica plate, let's imagine you're looking for an organism that's unable to grow on tryptophan. So you have this master plate, you've mutagenized all these cells, you diluted them just like you did for the enzyme assays and plated them out into individual colonies. And this is a minimal medium plus tryptophan plate. So everything will grow on it, including organisms that are unable to synthesize tryptophan. You then take it and you put it on this velveteen fabric, and the velveteen picks up a little bit of the culture on each of these little spots, and it's an exact pattern of what's on the plate. You then take the test plate, you plate it, you push an impression on there. Now these colonies, little bits of it get transferred, and this pattern here on the test plate is identical to the pattern on the master plate. You then incubate it, and then you look, and should say colony that didn't grow. So these are all colonies on here. They all grew. This colony didn't grow, and now you know that that colony that didn't grow on this master plate is trip minus, because this is a minimal medium plate, this test plate. So this colony didn't grow right here, and that one is unable to synthesize its own tryptophan. A third method you can use is color indicator plates. So again, you have a bacterial culture, you mutagenize it, you dilute it out, you plate it out. Now, this color indicator plate is a plate such that the desired property that you're interested in somehow lights up. It's different. You can observe it on a plate. So in this case, this is a McConkey plate. Imagine we're looking for lac minus cells. And these will be cells that will be, as shown on the photograph, they will be a lighter color than the rest of the cells that are dark red. So you can see that. So you just plate them out, you look through the plates very quickly, and you can find the lac minus mutant that you're interested in, pick it, as shown here, streak it out, collect it, grow it. These color indicator plates are a very useful way of doing things, because it's like the screen is done right on the plate. And in contrast to enzyme assay, where you have to assay every single colony, you can just look at the colony and find the ones that are doing what you want. Okay, color indicator plates. Finally, there's membranes and hybridization. Imagine you're looking for a specific base pair change, and sometimes you are. You can design a little DNA probe, as shown right here, that will detect that base pair change. So you take a membrane, you put it on this master plate, it picks up bits of colonies, you lyse the cells, you do a procedure where you add this probe, and then you can detect the probe in some way, sometimes with radioactivity, sometimes with other chemicals. And this colony that has this specific base pair change that you're looking for lights up. You can then pick that colony off of the plate and do whatever. 2 concept checks to finish up. A culture of E. coli is spread on a plate of LB plus rifampicin, an antibiotic. Colonies appear on the plate. This is a selection, screen, or neither. It is, in fact, a selection. The only thing that can go on the plate is the desired class of organism. Another concept check. You want to find mutants in histidine biosynthesis. A culture is diluted 1 millionfold and 100 microliters is spread on a minimal plate plus histidine. This is a selection, a screen, or neither. My guess is that you said screen, which is incorrect. The correct answer is neither. If you plate these organisms out on a plate of minimal plus histidine, all you're doing is supporting the growth of the organisms. Everything's going to be able to grow. Organisms that can synthesize everything that they need are going to be able to grow, and organisms that can synthesize everything they need but histidine are going to be able to grow. But it's not a selection, and it's not a screen. Colonies are taken from the previous minimal plus histidine plate and replica plated to a minimal plate. Those that don't grow are picked from the master plate for further study. This is a selection, screen, or neither. And the correct answer is this is a screen. You are screening for organisms that are no longer able to synthesize histidine. They could grow on a plate of minimal plus histidine, so they can do everything but when you put them on a plate of minimal, they no longer can grow. So they're mutants in histidine biosynthesis, and that is a screen. This brings us to the end of the show. I will see you in class.